From Microbe TV, this is Immune, episode number 18, recorded on March 26th, 2019. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back.
Where it must be getting warm, right?
Well, it's sunny. Let's put it that way.
It's not freezing anymore, is it?
It's sunny. It was 18 Fahrenheit this morning.
Overnight it still gets cold.
Yeah, but it's supposed to warm up, and I think ski season is probably over. But now we can look forward to springtime.
Also joining us from Durham, North Carolina, Steph Langle.
Hey there. Great to be back. I feel like I miss you guys after not talking for 4 weeks. Maybe we should do this more often. I don't know.
We really detached totally.
I guess we should. Yeah, we do.
And you get into your own thing and it's normal. Right. It's fine.
Well, maybe we can find ways to increase communication, and maybe we could I know we're all so busy, but I don't know. We can talk more about it.
Today, it's your turn, Steph.
It is my turn. Yes. I'm going to be talking about a paper that I found that actually has a connection with Vincent that we'll talk about later. But before I start talking about the paper, I was getting some feedback on the podcast, and there's some people that really want to engage more, but maybe are feeling overwhelmed by the content. So I think I'm going to start big. I'm going to talk about the big picture. I'm going to go smaller and smaller, and then hopefully come back out and get to the big picture, and people can tell me how they think it is, and hopefully engage maybe the non-immunologists in the audience.
Yeah, I think it's a good idea. Over on Twitter, we have a couple of people who have said it's too complicated. We don't understand it anymore.
Right. Well, I know. And but it is complicated, but I think when my friend told me this, I said, well, you know what? I just need to do a better job, because people can understand this stuff. It's definitely digestible, but I just need to do better. So that's my goal for the next couple months. So I want to talk about T cells. And in general, we talk about T cells mediating adaptive immunity. And they do this through very distinct antigen-specific contexts with target cells. So in an ideal world where we're healthy, things are functioning, if we develop something like a tumor or get infected with a virus or bacteria, it's our T cells that can migrate to the site of infection, or where the tumor's at, recognize malignant cells or infected cells as foreign and kill them. And basically repeat that process until hopefully the tumor is gone, or the tumor cells are gone, malignant cells rather, or the virus infected cells. That process allows our body to clear itself of potentially hazardous pathogens and cells. So
So I wanted to ask you a question, actually, because
sure.
you're a little bit generation behind me, but do you know whether or not we've always believed that the immune system could eliminate cancerous cells?
Well, I know that it definitely was not in fashion as it is now. For instance, there was not a time, I don't know how long ago, and you can maybe let me know, where we thought T cells or lymphocytes could clear malignant tumors. Is that
actually
correct?
Yes, but I think that there's a time when people thought that there wasn't immune surveillance. So it was in favor for a while, and then it went out of favor, and now it's back. I don't know. And Vincent, do you remember hearing anything about this kind of stuff? Because I remember being in graduate school, and people were not really enthused about immune surveillance and whether we could actually clear tumors. Because the argument is always like, well, you don't see them unless they escape the immune system. So there must be ways for the immune system to kill the tumors before they get too big. And then everybody was like, well, that's kind of a negative argument. How do you ever prove that?
Right.
So it was a difficult topic for a very long time. And it wasn't clear whether or not the immune system was capable of eliminating cancer. But now I think it's quite obvious that it is, but that there are impediments in place that the tumor cells themselves put up that inhibit or block or reduce immune responses. And so there's the great potential to harvest the immune system to attack tumors.
Yeah, and I would imagine that trajectory of what you just talked about has a lot to do with the technology that's available, sequencing tumors, being able to find mutations that result in mutagenic peptides that cells can recognize. And these things kind of contributed to, what we've talked about in previous episodes with CAR T cell therapy, and combination therapy. So yeah, at least in my graduate career, it has been, and especially now, something that people are interested in. Yeah, it's interesting what things kind of fade in favor and then come back once we, have better technology. So I'm going to relate it to Cd8 T cells. Now we know there are Cd4 T cells, but Cd8 T cells are cytotoxic T cells that when they migrate to the tumor or the infection through communication molecules called chemokines, which are soluble molecules that help kind of tell the T cells to come to the site of infection, or adhesion molecules, which helps the T cells roll along the walls of our blood vessels and transmigrate to the tissue. Once they're there, these T cells need to interact with something called major histocompatibility complex, or MHC. Now, all species have their own MHC. So for humans, it's called HLA. For swine, it's SLA. For mice, it's H2, I'm pretty sure, H2. And so what these molecules are doing is they're able to present an antigen on their surface when they are on the surface of antigen-presenting cells. And so the T cells interacting with the MHC molecule peptide complex is what allows them to become activated and then undergo their effector function, secreting cytotoxins, perforin, granzyme, which can form pores in these target cell membranes, and then the cells can undergo apoptosis. And so if you are picturing it, you have your T cell, which has a TCR, or a T cell receptor. The receptor then can bind to the antigen being presented by the MHC, which is on the surface of antigen presenting cells. So you have these like T cell effector cells, and then your target cells, or the cells that are going to bring they call an immunologic synapse together in order for the T cell to recognize the pathogen and then hopefully kill the cell that it's targeting. So within that process, both cancer and viral and bacterial infections can subvert that process. But what we're going to focus on, because it's the focus of the paper, is that process of T cell receptor recognition and MHC and the MHC peptide complex. And that recognition is really is obviously very important, because that's the whole basically how the T cell can activate and kill in a very antigen specific way. And the diversity within the T cell receptor is one thing. And then there's diversity in the peptide, which is a second thing. And then there's further diversity in the MHC molecule, which is the third thing. So the diversity of all these three things make it difficult to determine what the specificity of this T cell receptor is. And so if you can think about something like cancer, where you might not necessarily know what peptide is this cancer expressing, so that the T cell can see it, respond, and actually kill the cancer cells, it would be really beneficial if we could basically define what those T cell receptors are of an unknown antigen specificity. And so what they call that is called orphan TCRs. An orphan being that you don't know, one, what it binds to and what that cognate receptor is. I'll be saying that word a lot, and cognate just being, if you have a receptor, what does it bind to? What is the receptor that it binds to? So really, if we can find more TCR, T cell receptor ligands that activate T cells, the more likely it will be that we can engineer T cells to target specific cell types, like cancer cells, virus-infected cells. And so if you can identify these immunogenic peptides, that's kind of the way a lot of this field is going. You can not only design T cells, but you could vaccinate with an identified immunogenic peptide to increase the amount of T cell clones that recognize that peptide to then kill cells that express that peptide. So
I was thinking about this too. So one of the things that people have been doing for years is take a chunk of tumor and take the T cells out and then grow them in culture, because presumably those what we call tumor-associated lymphocytes or tumor-infiltrating lymphocytes, TILs, that if you expand them up and put them back in, they could then attack the tumor because they must be in the tumor because they're specific. And so I was trying to think of why identifying these specific receptors in a different way might be more advantageous. But I guess if you can clone them, you can make these CARs and other engineered, personalized medical reagents that would be, I guess, better than just expanding T cells in culture.
Yeah, I think it has to do with, one, the specificity. I think that there are the antigen you're looking for and potentially the reactive T cell that you're looking for maybe is one in how many T cells there are. And so potentially the process that they're doing now doesn't expand the clone they need at the capacity they need. It's not high throughput. You can't necessarily, I mean, if there are how many different mutagens, but the T cell only recognizes one or two that's effective at killing, the growing up the T cells may not be specific, high throughput enough. That's kind of what I got from reading and looking into all these different ways people are trying to identify T cell receptors that are reactive to neoantigens being antigens that are going to cause a response in the T cells to an effector response. So yeah, you're right. I mean, I think that was a way that could be effective, but maybe once they're seeing it just doesn't work with some cancers, they're thinking, okay, we need to be more specific. We need to be able to do a lot of these TCR identifications at once.
Yeah, that makes sense.
What fraction of maybe that's part of why they do this in this paper, but what fraction of peptides displayed on tumor cells are self versus neoantigens? Do we know?
That's a great question. I don't know. Cindy, do you?
I have no idea.
I mean, there's a lot of cross-reactivity. You know, I think what I can assume is that the amount of neoantigens where it does not cross-react with a self antigen is low. I just don't think it's high. I don't know a number, but I think it's pretty low. Yeah. So the name of the game then is determining these orphan TCR specificities. How can we, in a high throughput way, figure out what these T cell receptors are responding to so that we can develop technologies that select for that, and then we can develop future technologies in terms of, like I mentioned, vaccinating, CAR T cells, things like that. So there have, as Cindy mentioned, been a number of strategies that people have used to determine orphan TCR specificity. There's a couple I'll go through because they're just interesting, but mass spectrometry has, I think there was the paper I'm thinking of was in 2014 or 15. And basically when you purify the MHC, which is on your target cell, your antigen presenting cell, the peptide complex, and eluting out the bound peptide and using mass spectrometry and bioinformatics, you can analyze all the peptides from all the extracted purified MHCs and compare it to healthy tissue, to a tumor, and in that way potentially discover these neoantigens. The problem with that is you need a large number of cells to be able to extract this peptidome, as they describe it in these papers. And mass spectrometry really, I mean, it is going to detect more abundant peptides. So it's going to favor ones that are more similar. And that's really not what we're looking for, right? We're looking for ones that are underrepresented or subclones, like malignant subclones that have mutated on the tumor. So it's not an ideal method, but it is one way that people can look for these orphan TCRs. Another way, and it's kind of integrated through all of these, is determining them empirically. And so if you sequence tumors, identify mutations, you can use algorithms and software to predict if that epitope is going to be immunogenic. And then you develop a synthetic peptide, and then you can do T cell proliferation studies to see if that peptide actually causes the T cell to proliferate, be cytotoxic. But again, I mean, you still need information about the target to identify if you can then make the synthetic peptide. So it's lengthy, again, cumbersome. So it's interesting, but maybe not ideal. Now, some of the technologies they use in this upcoming paper I'll talk about are called Peptide Mhc Multimers. And basically, if you can generate synthetically MHC complexes with your peptide of interest, it can help stabilize the MHC peptide on the surface of the cell. You could then put a fluorochrome on that peptide MHC multimer and look at through flow cytometry to see if there's any antigen specificity to the T cells. And there was an interesting paper, I think it was last year, where they're using these yeast display libraries of surface displayed MHC complexes. So you generate MHC peptide complexes and then you express them on the surface of yeast cells. And you do this for many different types of peptides. And you have your yeast cells, you co-culture them with these beads that have the TCR on the surface, and you see which ones interact. And then from there, you can sequence and see, okay, which ones are interacting. Then you can do some functional studies after that. So all of these technologies, you want to rapidly and robustly identify ligands, but you want to be able to do it without knowing information beforehand. You want to be able to just take a tumor, try to identify what the neoantigens are, and then be able to very quickly determine if a T cell can respond to that. And so that's where this paper, in terms of the big picture, what we're talking about. So with that, what this paper took advantage of is something called, and I love this word, trogocytosis. Now, in Greek, trogo means, I remember it being like to gnaw, but I've seen, I think, like nibble. To nibble is another way to describe it. And it was described it had been described in like the 1970s, 1980s, but the term trogocytosis wasn't coined until 2001. And basically what this means is that you have cells that are transferring their own membrane patches to a cell that they're in contact with. So think about what I just talked about in terms of the MHC holding that peptide on the surface of an antigen-presenting cell. When it comes in contact with the TCR in that immunological synapse, they have to touch. They're conjugated together. And in that process, there is actually transfer of surface membranes that are not, or surface membrane proteins that are not necessarily just the peptide, but could be the TCR receptor itself going to the APC. It could be the other way around, where the APC transfers part of its surface membrane to the T cell. And so it's that intimate interaction where they fuse and they separate and they've acquired a piece of each other. It's pretty cool. I've included a YouTube video. So if you, Vincent, you can put that in the show notes, and you can see videos of cells coming together where they've stained dyes, and the one cell will, after it leaves the dye from its cognate cell, it will retain some of that as it migrates away. So it's a pretty neat phenomenon, and I think it works in a couple different ways relevant clinically, both basic and translational science. Cindy, when you teach trogocytosis, do you teach it within the context of like T cells, or are you teaching it in a different way?
So we don't usually talk about it much.
It seems to be trogocytosis.
Should talk about it more.
Well, it seems to me, looking at the literature, it's not, it pops up here and there. It's kind of like a so what until you can see if actually, it's used for something, kind of what we're going to talk about in the upcoming paper.
So I know one of the ways I've heard of this happening is related to cancer as well. But so when you're using immunotherapeutic antibodies, cells that have Fc receptors that can bind to those antibodies will sometimes yank on them. And
yes.
The antibodies will come out and they will pull bits of the membrane from the target cell with it. And so there's a way, and I don't know exactly which cells are involved, but when you have a B-cell lymphoma and you're trying to use an anti-CD20 antibody, immunotherapeutic antibody, to remove those cells from the bloodstream because they're aberrant, they're growing out of control, that sometimes the whatever cell is going to bind to that and pull that CD20 off allows that cell to then survive. Actually, it's a bad thing because if the antibody comes off and they strip off the CD20s off the B cell, now it's no longer a target for the anti-CD20 therapy. So there are ways that it's involved in resistance as well. So I think that this is a really interesting idea here, that they're using this whole thing as a way to map, as a tool. I think that's a really cool aspect of this.
Definitely. Yeah. So your example is one that I found fascinating, because you spend all this money and this time to have somebody get rituximab, which is the anti-CD20 antibody, and then you have, tumor cells that are essentially stealing it from the surface of your B cells, I'm sorry, FCR receptor positive monocytes, rather, stealing it from B cells. Yes. And I heard, so in these papers, they kind of refer to it as shaving instead of, they say trogocytosis, but they refer to it as shaving off of the B cell, the antibody rather. So there's a couple of different ways that it's described in the literature. It can be bidirectional, where both the APC and the T cell take parts of each other, or it could just be unidirectional, where you just have, let's say, the target cell, the antigen presenting cell, taking co-stimulatory molecules or adhesion molecules from the T cell.
And I wanted to stop for a second, because I know I keep interrupting, but
good. I love it. It's great.
Boggles my mind that the strength of the interaction between whatever two contact molecules are is stronger than the membrane remaining attached to the cell, right?
Right.
Physics wise, this just astounds me that it can work that way.
Well, and they described that the microvilli that are on the surface of the T cell, and that they can help produce these nanotubes where you can actually exchange membrane organelles in between different cells. But these microvilli on the surface are actually fragile to extraction. So when it's the force of when the two cells separate, there are just bits and pieces. But it does seem that in some papers, they describe actual enzymatic cleavage, that it's an active process and not just a passive process where it's just like accidentally extracting because it's fragile. So yeah, I agree. It's really fascinating. And the implications clinically, one of the other examples was activated T cells in the area, in a microenvironment of a tumor, acquiring a MHC or a HLA that's associated with the tumor. It will trogocytose it on the surface of the NK cell, but then it will stop proliferating. It will no longer be cytotoxic, and it itself will act as a suppressor cell to inhibit other NK cells. And so essentially, it's a tumor, it's a cancer tumor-induced immunosuppression of the cells itself from trogocytosis. So fascinating. And it can be involved in pathogenic infections. So there's a lot of different examples where and recent examples where it was a way for bacteria to spread cell to cell without actually having to go in between cells, without having to maybe butt off into vesicles. It could just be you blip from one surface to the other. And the other way was there was a parasite. So this was a way that neutrophils kind of nibbling, or they describe it as taking bites. It's very silly, from a parasite to induce death of the parasite. So it works both ways, almost, that, the function of this phenomenon is to protect cells, that if it can potentially apoptose parts and bits by doing this, or the opposite, where something pathogenic can subvert this, what's going on. So really interesting. I think possibly we brought it up in our last episode, and I wanted to talk about it. And this was the paper that I thought was interesting, and why this is happening. If you think about an ability of a cell to retain pieces of a membrane, you could be signaling even after you've done, you've stopped interacting with that cell. It's kind of like sustained cellular signaling after they've unconjugated, which is interesting. And yes, so that's the introduction. Anything I could help clarify from you guys?
Does this happen with certain cell types only, or can any two cells that engage in a ligand receptor interaction do this?
From what I understand, both from the paper we're going to talk about and from reading, it is an antigen specific process. So it has to be cells that are coming together and can bind each other's receptors and ligands. I don't think this happens as much as just cells that maybe brush by each other. This is definitely antigen specific. So,
but if I took, say, two cell lines and made sure there was a ligand receptor interaction possible, would they be able to do this?
I don't know. I would say yes. A lot of these examples, I think it would be interesting to then stain the cognate cell line with antigens on the surface of the one you added to see if you see any type of, membrane on the other one. But, okay, cool. So trogocytosis is an interesting concept, but how could we use trogocytosis to identify orphan T cell receptors, as I described before, is a problem and something people are trying to work on as we are ushered into this new era of immunotherapy for cancer and trying to be more specific with vaccine development. So the paper title is T cell antigen discovery via trogocytosis. So pretty easy. There's co-first authors, Gwendang Li and Michael Bethune, and it's out of the David Baltimore's lab at Caltech. Now, Vincent, David Baltimore, you did your postdoc?
I did.
Excellent.
Know him quite well, yeah. Just wonderful time, and I saw him last year at his 80th birthday party.
Wow. So he's still pumping out very, important work at 80. So that's, motivating, I would say.
Yeah, it's amazing.
Awesome.
Although he's retired, he's closing his lab soon.
Oh, is he?
Okay.
What he said in May was that in a year he would close it down. So coming soon, he's going to close it down for sure, yeah.
Wow.
How many people were at that 80th birthday? Was it hundreds and hundreds?
Oh my gosh. First of all, there were 200-300 people.
Wow.
But not just, in every field there were luminaries. These are people that worked with him. And Harold Varmus, Eric Lander, Bob Weinberg, Phil Sharp. And on and on and on and on. It was just amazing. And then I was there too.
You see, you're a luminary, please.
But it was fun.
Cool.
It was great. It was great science, and it was a good time.
So did they have a symposium?
Yeah, they had an all-day symposium of talks. It was great. And they had some panel discussions, which were pretty interesting. They had one with leaders, right? So there's a lot of science, which was great. But then they had leaders where they had a president of a medical school, Mark Schlissel, president of University of Michigan. Actually, not a medical school, a university. And they had a CEO of a company, all these people who got leadership positions, and they talked about why they did that and what was different about science and so forth. So it was a really, really good day symposium. And then there was a dinner at night, which was also very good. That was great.
So when you were in his lab, was he interested in T cell receptors and T cell biology at that time, or is this something that happened later?
So it happened later. So when I was in his lab, I was in the polio group, but he had a retrovirus group, and they got interested in immunology because of Abelson murine leukemia virus, whose oncogene has immune functions, and that got him really interested in it. But at the time, he was just, I'll tell you, when I got there, they were just starting to clone immunoglobulins.
Wow. Right.
And to find out all about them. And from that, other things developed. And his immunology interest got bigger and bigger over the years. And now supersede really his virus interests completely.
Interesting. Well,
yes, very interesting.
That's great. So with this paper, what their goal was to develop a TCR ligand discovery platform by exploiting this trogocytosis phenomenon we talked about in a way to mark target cells that are presenting a peptide MHC cognate, and that they are going to be marked by the TCR on the T cells. And so what that's supposed to do is, if these two cell types with these they're presenting two different types of receptors and ligands, if they come together and they come apart, we can look at them and say, okay, these for one bound together, and they bound strong enough that the T cell could potentially become, undergo its effector function. So we can select for that peptide that T cell reacted to. So that's the goal. So what they did was they took cell lines and they expressed T cell receptor antigen pairs. So if you hold up your hands, and in one hand you have something called a Jurkat cell, and in the other hand you have something called K562 cells. The Jurkat cells are a cell line, it's an immortalized cell line of human T lymphocytes, and the K562 cells are basically they're your antigen presenting cells. They're hematopoietic stem cell, or cell that comes from a person who had, I think, leukemia. Yeah, chronic myeloid leukemia patient. And so this malignant cell can spontaneously differentiate into antigen presenting cells. That's basically the cell type that they're interested in. And so the Jurkat cell, the T cell, they engineered it to express one of two TCRs, either something called F5 TCR or 1G4 TCR. All you need to know is that T cell expressing one of those two TCRs are specific for two different types of cancer antigens. So F5 is specific to recognize something called MART1, which is on melanocytes. So it's a melanocytic tumor, melanoma, essentially. So this TCR can recognize that protein on the surface of melanomas. And 1G4, I'm not going to probably I'm not going to reference this NY-ESO, but it is a human cancer testis antigen. So melanocyte antigen, cancer testis antigen. These two T cells, cell lines are going to express that TCR. And they then engineered the K562 cell to express something called a single chain trimer. And these single chain trimers are essentially, they are MHCs complexed to the cognate peptide. So if the TCR can recognize MART1, the single chain trimer is an MHC presenting MART1. So that's the whole goal, right? You have T cells that can respond to target cells presenting the MART1, which is the melanoma tumor antigen or the human cancer testis antigen. And what's interesting about these single chain trimers, or SCTs as they're referred to in the paper, the way that they link these single chain trimers together allows them to be presented on the surface of the K562 in a very stable way. So it's kind of a super physiological expression of this peptide. Basically, a normal antigen-presenting cell probably isn't going to express this MHC peptide complex with such stability and at such high levels, but it allows you to, obviously, if it's at high levels, you know it's going to, if the T cell is there to respond to it, it's going to bind to this single chain trimer.
So I was thinking of how one could explain this, so we always talk about, at least I do, MHC binding peptide as a hot dog in a bun.
Yes.
And so I sort of thought, well, this is a hot dog in a bun and the hot dog's chained to the bun. So
yes.
It stays in place. It's a very short chain. So it stays in place so your hot dog doesn't slip out the end of the bun when you're trying to eat it.
That's a really, really good way to describe it. And looking at this little picture I've included, I mean, it looks just like that. The linker is anchoring the peptide down, and so it can stably express on the surface of K562. So it's a good way to engineer these two types of cells to then do what they did in the upcoming experiments.
And the other thing is it's called a single chain trimer, and people might be scratching their head because we're saying MHC and peptide, like two things. But for those who are a little bit more savvy here, the MHC that we're using is called MHC class 1, and it's a dimer of something called beta-2 microglobulin, and then another chain of MHC. So it's the beta-2 microglobulin, the MHC, and the peptide. So there's actually three things in this, and they're all fused together so that they don't fall apart.
Right. So if you transduce a K562 cell to express this, it's going to have all the components that the MHC1 molecule, it's mimicking the native MHC1 molecule.
Exactly.
So what they did was they took their cell lines and they first wanted to see, upon antigen-specific interaction, can they experience trogocytosis? And so the first thing that they did was they wanted to look at just non-specific transfer between the two cell types. And so they just labeled with something called N-hydroxylsuccinimide, which labels the membrane of the cell. So it's not specific to any antigen. And they labeled, and it hooked with a biotin. And then the biotin, you can add streptavidin with a fluorochrome to help identify the cells. And when they co-incubated the Jurkat cells expressing the TCRs specific for the peptide presented by the single chain trimer on the K562s, they were able to see that there was some non-specific transfer of biotin. And I think why they did that was they wanted, and maybe Vincent, this answers your question, that's a non-specific interaction, that biotin, and they did see a shift. So I think it means that as the cells bump into each other, that there may be some type of transfer of molecules on the surface. But when they had that nonspecific shift be their baseline, and then they went ahead and incubated together the Jurkat cells expressing the like, let's say the cancer testis antigen with the K562s that presented the cancer testis antigen, there was a huge shift, a 3.5-fold shift over the nonspecific shift. So what they're telling us is that's demonstrating an antigen-specific cell trogocytosis. And they're doing this because all these cells are labeled. They can see if the target cell, the K562, is expressing the TCR, right? K562s, they should not be expressing TCRs, but if you stain them with an antibody for TCR and it shows up, that means they underwent trogocytosis. So that's what they're doing, co-incubating and seeing if these cell surface membranes are ending up on the other cell type. And they're seeing that it is happening in a cell-specific manner because when they co-incubate non-cognate T cells and target cells, they do not see membranes ending up on the target cells. They do not see a shift in TCR. So they went through a bunch of these experiments, basically trying to answer a couple questions. Does trogocytosis happen? Is trogocytosis specific to surface receptors only, which they did show? So intracellular receptors, the target cells express a green fluorescence. They did not see that green fluorescence on the surface of either of the cells. So it was only surface receptors that could transfer from one cell to the other. A couple of them were not only ones included in that immunological synapse like MHC. They also looked at things like the T cell expressed something called low affinity growth factor receptor, and it's not a part of that immunological synapse. And I think the purpose of that was to show this can be the transfer of membrane proteins is not just within that MHC TCR complex, which is interesting. So that is looking at some of these beginning studies. And then they also want to see, is it dose dependent? And so they increase the amount of peptide that they had on the single chain trimer on the K562 cells and saw that trogocytosis of the TCR transferring to the target cell increased with more peptide. They also looked to see if things like Cd8, which is specific for MHC1, if that enhances the ability of trogocytosis, and it does, which makes sense considering Cd8 is a co-stimulatory molecule within this interaction. And so they go through these experiments just trying to show to you that it's happening, and that it's not only happening in a specific way. I think one of the things that they were worried about, which is that because these single chain trimers present kind of this supraphysiological expression of the peptide, can we add different variants of the peptide and see if they respond? And so they co-cultured the T cells and the target cells with different variants of the melanoma and TESI antigens and found that they responded differently. So why they focus on that, I think, is to show it's responsive to mutations, which is important when we're looking at cancer. Or anything, cancers. So you're going to have a high amount of mutation, which is kind of the whole point of identifying these TCRs. And so with that, they thought, well, if we're showing that trogocytosis is happening by flow cytometry, by dose-dependent experiments. So let's see how sensitive the system is.
I really like this experiment.
Yeah. Because it kind of gets past like, okay, we know it happens. It was good. They had to do that to make sure that like a proof of concept that it was working, but okay, let's see how sensitive this system is. So what they did was they took they had these target cells are green fluorescently expressing cells that have this human testis antigen peptide MHC complex on their surface. And they co-cultured them at a 1 to 10,000 ratio with K562 cells that are not expressing the human cancer testis antigen. And so that's quite a bit, right? I think the purpose of the 1 to 10,000 is to say, okay, is it going to be sensitive enough that when we add these JERCAT T cells, can they find each other, basically?
Can you find the needle in the haystack, right?
Yeah, exactly. Because if this isn't sensitive, we're not going to be able to detect the small amount of reactive T cell receptors in something like a tumor. So that was the point of this experiment. And so when they mixed ones that were positive for the antigen and ones that were not, and they added the T cells that were engineered to express the TCRs, they found that only when you incubated the ones that were the cognate receptors, so only when you incubated T cells expressing let's say the TCR for the melanoma antigen, incubated them with the K562s expressing the antigen, you saw trogocytosis. So they did find each other. So that was the point of that, was to determine how sensitive it could be. Now, 1 to 10,000, is that biologically relevant? Could we increase, what about 1 to 100,000 or 1 million? I don't know if they chose 1 to 10,000 based on some type of standard curve. It'd be interesting to know why just 1 to 10,000.
I don't know. By flow cytometry, looking at the data, I don't know if you could have gone any more than that, because they're already pulling out. I don't know if you've noticed that 0.009% of the total events. That's
right.
Wow. Like I don't think I've ever done flow cytometry where we're pulling out anything less than like 0.1%. That's so already
probably just this
really,
sensitivity.
This is amazing that they can pull anything out.
It is amazing. Yeah, that they're able to find such low, rare populations of cells. So you're right. It may just be a function of the sensitivity of how they're detecting those positive cells. Yeah.
But I think 1 in 10,000 is still pretty darn good.
Yeah. No.
But they didn't try any more dilute, because they say they made this.
Right. They don't present data.
But they don't say we tried 1 in 100,000,
right. No.
It didn't work. Or whatever. Yeah.
And the other thing I don't remember seeing, and maybe if you guys saw it, but I know the mixture of the T cells was described, but I don't know if they, how many of those cells were added. I'd have to go back and see. Because the proportion of T cells with, if you increase the amount of T cells, that increases the likelihood of finding the target cells. And so that might not, it might kind of bias your assay if you're just having a huge amount of T cells, and they can find that one K562 cell. So I don't know, but anywho, so yeah, that's basically determining that their assay is sensitive. So they move on and they want to see, okay, is it specific? So from this point, we've really only been looking at these F5 TCR and the 1G4, the melanoma and human cancer testis antigen engineered TCRs. Now, if we made a whole library of many, they use 12,000 different T cell epitopes, and then we add the transduced target cells, can they find that one F5 TCR within 12,000 epitopes? And so what they did was they generated a cDNA library using public T cell epitopes from the Immune Epitope Database. And because T cells only recognize 8 to 12 amino acids, that's how they selected the length of the epitopes. And they created a cDNA library of single chain trimers. So essentially you have all of these different MHC peptide complexes that are being transduced into T cells. And so they're expressing thousands and thousands of epitopes.
How does this seems like an incredible amount of work.
So, I know how you make a cDNA library, but,
But this is with specific, I think you would have to order each sequence individually, right? How would you do this? Because it's not like you're just making a general library from something that's out there. Like if you're going to make a library from a group of cells that you collect, that's fine. But this is like specific sequences that they're putting in a library. I think they may have to make each one of these individually.
So yeah, I think so. Wow. And probably that's why it's in Nature Medicine, maybe. Because it is so, it's a huge amount of epitopes that they're engineering to then be presented, on these single chain trimers. So it's a huge amount of work and definitely should be commended, because they describe other technologies, and the amount of epitopes they're able to do with those technologies are 3,000 epitopes, or, a couple hundred different, peptide MHC complexes. So yeah, this was definitely kind of a brute force way to do this. So what they did then was similar to other experiments, they co-incubated those K562 cells expressing the SCT library, and they co-incubated them with T cells. Now they kept their F5 and 1G4, the cancer antigen, as positive controls for this assay. So, they want to be able to for the Jurkat cells to still find within the sea of expressing K562s if they can still find each other. And so they co-incubated, and surely enough, what they found was they were able to see trogocytosis only when they were cultured with the cognate antigen on the T cells. And so it is specific. They were able to find each other within all these different epitopes. And they go through and they identify a couple of the ligands, and this is working towards what their goal is, right? To identify specific ligands that they can, make a vaccine or basically you're just trying to enhance the ability of the T cells to recognize that and kill an infected cell. And so they kind of list them there. I think for the MART, for the melanoma antigen, there were more, 8 out of the 9 that were most represented in the population of T cells that they were able to interact with, the K562s. And then in the human cancer antigen, there's only 2. So I think it's just a function of that antigen. But they were able to identify specific ligands from this assay. So they found out it's sensitive. They found the cells were able to find each other within a huge amount of K562 cells. They found out it was specific, that the T cells were able to find their cognate receptor in a sea of MHC peptide complexes on K562s. And so the next step then is, we need to be blinded. We need to be able to, this platform needs to be able to find ligands that are able to bind to T cells without knowing much about them. Because that's really the whole goal of this type of orphan TCR ligand discovery. So they went through a process that they don't really describe in the methods because they say unpublished data. So I think how they did this, it doesn't really describe it totally. But they basically they were able to do exome and RNA sequencing of a tumor material from a person with metastatic melanoma and use predictions of which of these mutations is going to generate an epitope ligand that can be presented on the MHC and reactive to a TCR and a T cell. And so, I'm assuming that when they were able to find, they were using some algorithms and potentially software to be able to predict, okay, this mutation is going to result in a very immunogenic peptide. I don't know if you all have any experience with any of this, if you had any comments.
No, but we definitely talk about anchor peptides, right? So for each HLA, we using technologies that you discussed earlier, where you elute all the peptides from a particular MHC molecule, and then you sequence them, you can find what are the anchor residues. So those residues which are absolutely required for any peptide to bind that particular MHC molecule. And so they can use those known reference aspects of what the primary structure of a peptide will look like that's capable of binding to that. And so I think also with that information, if you look at what the non-MHC binding residues are that are going to be facing up towards the T cell receptor, some have more structure than others. And so they probably are more likely to create a strong interaction with a T cell. So I think that you're right. There are, based on many, many, many bits of information we know for each MHC molecule, algorithms that can predict which will be peptides that will bind very effectively to that MHC molecule and which of those will probably be strongly immunogenic.
Right. And so, and I think that they use that and they design kind of these multimer panels to isolate reactive T cell receptors. And so by doing that, they generated another single chain trimer library, this time consisting of only 3,000 unique epitopes. So a little bit less than the 12,000 from earlier. And they transduced the K562s with this library and then co-incubated and then they were able to transduce the T cells with the TCR receptor that would respond. So that's the thing. You need both in this case. Before we were only talking about maybe transducing one with a library, but here you're transducing the K562 cells to present this neo epitope, and then you have to transduce the Jurkat cells to express the neoantigen TCR. And so which one is responsive to which, and they do co-incubation experiments, and they're able to find 5 ligands that, after selecting for trogocytosis positive, so basically the target cells expressing the TCR, which it shouldn't unless there's trogocytosis, they found overlapping mutated peptides, and that peptide is called ubiquitin-specific peptidase 7. And so they then took that information, they took that peptide, the USP7, and then they further transduced it into the K562s to see, okay, let's do this by itself now. Let's take it away from all the 3,000 unique neoepitope expressing K562s. And they saw that there was interaction with the this ubiquitin specific peptidase 7 expressing peptide MHC complex with the TCR on the T cells. And so what they were able to show is that you can take a tumor that you don't know a lot about, you can predict information about the mutated peptides. If you then transduce these T cells and antigen presenting cells, how they interact, you can determine if they are by trogocytosis, if they are interacting, and then you can use that information to say, okay, this is a ligand that a TCR will respond to. And that was kind of like the big, last experiment, because we don't always have the information. So it needs to be blinded. It needs to be unbiased in terms of selecting for these ligands. So that was, go ahead, Cindy.
No, so I was going to say, so let's break this down just a little bit more. And I know you're going to go back out to the big picture, but so if someone has, if you've got a patient that has a tumor, the idea here is we want to find out what antigens the tumor is expressing and what T cells the patient has that can attack those tumors, so that you can try and engineer the T cells or T cell receptors to be strongly reactive against a tumor, so that if you put them back into the patient, they'll be able to attack it.
Yes.
So in reality, we need to sort of clone both of those things like they did in this last experiment.
Mm-hmm. Right.
So it's not a trivial undertaking to try and think about translating this into a therapy, because you're going to have to take a chunk of tumor. You're going to have to sequence that tumor. You're going to have to run it through an algorithm, find the neoantigens that are potentially active, clone them into a target cell. Then you're going to have to take the T cells that you purified at the same time, and then you have to incubate them together and then see which of your, readout cells now become TCR positive, and then go back and try and find out what that particular peptide was. And now use that peptide to try and maybe immunize the person so that they make lots of T cells that recognize that, so the T cells can go into the tumor.
That is an excellent summary. And I think that is the exact challenge of this type of work. And you're right. I don't know, that was one of the things I had asked, after doing all of that. I wonder, they say it's high throughput. I guess it is faster than the other technologies. We'd have to time them, but it would still take quite a while.
Yeah.
Sure.
So after a certain point of looking at enough people and enough different tumors, would you reach saturation where you say, here's this kind of tumor, we know exactly what peptides?
I think it's a great question.
It can't be infinite, right?
No, but I think I think each tumor has its so here's the rub, right? Each tumor has its own individual mutated profile or signature, right? And that isn't even constant. Right. And so there are what we call stem cancer cells, and the proliferating ones. And so they proliferate at different rates, and they have different levels of mutations in them. And so if you metastasize, you might have different mutations that you acquire in addition to the ones that were in the primary tumor. So yes, we could eventually exhaust potential mutations, but isn't that really the entire proteome that could be mutated? I mean, It's not that big because, for example, there are gatekeeper mutations that are really common, P53, P21, things like this that are involved in proliferation. If you mutate them, most cancer cells will have that particular mutation. And so if within that P53 gene, which is a common gatekeeper mutation, so once you mutate that gene, now all of a sudden you start accumulating more mutations because of the fact that cells are proliferating when they shouldn't because they have mistakes. Though the mutations in that particular gene are probably reasonably characteristic. You can't mutate any part of a gene and have it function like a tumor suppressor or a tumor activator mutation. There's certain mutations that are much more frequent. So yes, to a point, I think the neoepitopes could be identified. Could you then use those as a vaccine rather than trying to purify them this way? I'm not sure. I just felt like as I got through this whole thing, I'm like, that's so cool. How the heck are they going to do that for real? Right?
Yeah. Right. And I think, the other thing is that in an individual, there are multiple MHC or HLA molecules with heterozygosity and so these different alleles, it's a wide variety of peptides that are going to be presented. And so in a population, when you have all these different individuals with varied MHC alleles, the capacity to respond to a tumor becomes a lot greater in terms of complexity. And so I think we could narrow down maybe a tumor type and what you were saying, Cindy, these gatekeeper mutations, but then considering, this paper only looks at HLA-A2 restricted TCRs, so ones that are presented from Cd8 T cells to MHC1 molecules, and the A2 gene is a certain population of people. So that's not even considering different ethnicities. That's not, so there's I think a lot to consider. Of course, it's worth doing because, we're not going to this is kind of the new frontier, but I think it's going to take a while, but I love the concept of using a biological phenomenon like trogocytosis. There is a paper published in pair with this one from the same group where they're looking at dendritic, I think it was dendritic cell, but antigen presenting cell receptors that can help you can basically do the same type of process of looking for ligands. So that's really what CRISPR did, right? It's taking advantage of something already going on in the cell for, whatever you need to get done. So Cindy, your broad overview, I could not do a better job than that. So I'm going to leave it at that. I'm not going to try to up top that because that was excellent. But I think, one thing that I found, there wasn't a lot to criticize. You could say, well, what about Cd8 T cells? And they even bring that up. They think that this can be, extrapolated to Cd8 T cells, to different types of MHC2 molecule or MHC molecules. But they do, the first thing in their discussion was they say their key discovery is that target cells presenting superphysiological levels of peptide, MHC complex to T cells, this interaction causes trogocytosis in an antigen specific way. I don't think, a key discovery that was already known in my opinion.
I know. I read the same thing and I thought,
really? You didn't discover trogocytosis, but their next sentence is what I think is the key discovery is how can you exploit this to mark ligand presenting target cells with neo TCRs that you don't know. So I think that's kind of more what this paper brings in terms of the novelty.
Yeah. And I love what you said though about how, and it's the idea of the exploiting, like CRISPR, where you take something that you find as a biologic phenomenon and you say, how can we use this?
Exactly.
And I think that is the really creative thing that we can challenge the graduate students and postdocs out there who might listen to this is think about your system. What are you learning from that system? And can we use it? Not just understand it, but use it.
Yeah. And we talked about that with the Nobel Prize.
Exactly.
With James Allison, he was just interested in T cell biology. And Vincent, you bring this up poignantly. It's, if we limit kind of these basic research questions about trogocytosis, which otherwise, who cares? But it, how can we use this? So yeah, I agree. Any other thoughts on our paper?
Yeah, we were doing a paper the other day and we got to a point where we we said, we're not sure why this problem should be done, but it should be done because you never know.
Yeah,
exactly.
Right? Sometimes you can't know why you should solve a certain problem in science, but you should always do it because you just never know where it's going to go.
If it's a good problem and it's done well, right, with solid reproducible data, I think that it's worth doing.
Yeah,
for sure.
Because you don't know who will read it. I don't know that I would have ever come across this paper because it's not something that I normally work on. But I have.
Nope.
You see it and you're like, huh, and your brain just thinks in a different way, and you're like, wow, I would have never considered thinking about it that way. And so it broadens your mind. And so it's even that aspect of it where those of us who are in science, the more things you can read that are sort of that open up your mind of how to think could trigger something in you where you make a discovery that you wouldn't have otherwise. So we just don't know how, because everything is just an interconnected series of web and events and thoughts and ideas.
Yeah. Yep. And I think, this type of stuff, like Cindy, what you're talking about, it's how exciting that potential that there's something there to be discovered. I mean, that's what gets us through, kind of the other things that we have to deal with in science in terms of
labeling tubes and pipetting and
and grant funding and, people being weird, all that stuff.
I want to point out one of the co-authors, Owen Whitty.
Oh, I know that name. Yeah.
So Owen was a postdoc with David about the time I was there at MIT as well. And then Owen got a job at UCLA, which is where he is now. He's been ever since.
Oh, great.
But he's been quite a good, productive investigator. I remember Owen because he'd come in the morning, and in his shirt pocket, he'd have a list of things he needed to do that day. And as he did them, he would check them off.
It's a good way to go about it.
And I've never seen anyone do that. I certainly do not make lists. I probably should, but I don't. I don't make lists.
I was listening to you, and I'm like, that doesn't sound unusual to me. Where's this story going?
I don't know. You could. I just prefer to keep things in my head, which isn't perfect, but the assumption is all that you need to do, and sometimes you don't. So it's not on your list. If you're driven by the list, it can be a problem.
That is
anyway,
true.
It was funny. Everyone made fun of him, but he's been productive, so don't make fun of anything.
There's nothing wrong with lists.
Right.
Nope. Let's do 3 emails before we do our picks,
great.
Okay? Because we have a bunch that are accumulating. The first is from Emily. Hello, I enjoy your podcast series. I was just listening to Immune episode 10. It's a long time ago. Was excited to hear the mention of faculty member Vera Tarakanova from the school I attend. I did want to clarify, though, that our name is actually the Medical College of Wisconsin. Steph referred to us as Milwaukee Medical College. And since we are currently working on some rebranding, I thought it important to clear that up in case you'd like to give us a shout out in the future for any reason. Thanks again for the great podcast series. Emily is a MSTP, MD PhD student at the Medical College of Wisconsin.
Great.
Yes, of
I'll
course.
keep that in mind.
It is. Yeah, Vera was on a TWIV episode that I recorded.
Yeah, she's great. She did at last year's American ASV conference. I was in a group with her where she was doing kind of like a small session with grad students and postdocs, and she's awesome.
Steph, you want to take the next one?
Sure. Ross writes, hello, I really enjoyed the episode 14, Breaking Bad. A reference was made to a podcast with respect to cost recovery to the government NIH funded research. I was unable to locate that link. Could this be provided? Thanks again for this podcast. And he's referring to minute 47 in episode 14. And he says to you, I guess you referenced a Freakonomics episode you couldn't remember. And so he put that in there.
Yeah. So it's Mariana Mazzucato. It's Freakonomics number 348. And I think
you
put that in. I'm
too,
sorry.
Steph.
Yes.
You wanted to know that as well,
Mm-hmm.
Steph. That's a really good one. She talks an interesting way about NIH and its research and support and all that. So check it out. Okay, one more. Cindy, can you read that?
Can I skip one?
Yeah, you can skip it, sure.
Can we do
that's fine. We'll do that
We'll
next
do
time. Aditya.
Aditya. So we can do Aditya? Okay. Yep. So Aditya writes,
That's why you wanted to do that one. Right?
I just listened to the latest episode of Immune today, and I'm so grateful for being the winner. On a side note, I'd like to mention how listening to Immune, TWIP, and TWIV convinced me to study biology this year. More broadly, how my love for programming and computer science got me fascinated with all things microbiology. I loved studying computer science last year, but there just wasn't anything like it on offer this year. So the next best thing was studying a subject called student-directed inquiry, where I spend a subject line to complete my own research project. Doing a little bit of Googling and deliberation, I thought, hey, computational biology sounds like a really cool field. I'm going to learn more about it. Then over the next few months, I just got more and more fascinated by the natural sciences, and I came upon your podcasts. It really changed my perceptions about what biologists actually did all day. I thought it was a soft science. It was pretty observational. It never occurred to me that biology, just like everything else in this world, involved mathematics, chemistry, and physics, or that it could be computed. My inquiry doesn't involve bioinformatics anymore, and instead I'm studying how CRISPR and other gene editing systems are used in medical research and therapy. Alongside my inquiry, I'm also studying chemistry, biology, and specialized mathematics. Wow. I've come to love my biology and chemistry lessons very dearly, and I tentatively plan to study something like biochemistry or bioinformatics in university. I just can't get enough of it all. Sorry for the very long email, but I'd like to thank you again for selecting me as the winner for that textbook. It really means a lot. Aditya. P.S. If any of you are interested in an email interview about CRISPR or could recommend anyone who's involved with CRISPR, it would be really appreciated. Now, as I was reading this, and I'm thinking, this is not a college student. This is a high school student?
No, high school. So imagine
wow.
high school figuring out that you want to do computational biology. I mean,
wow.
Considering how many job postings I see for computational biologists, I mean, that's
Go
great.
for it. What I'm just amazed by is if you have found that love for learning and you found something that lights that fire in you, that's amazing that you did that early and you haven't decided what you're doing in university yet. Go
Definitely.
for it. Congratulations. And keep up the amazing work. All
right. Aditya, I interviewed Sam Sternberg for TWIM, and he is here. He's he worked with Jennifer Doudna on CRISPR, so you might want to email him. You can find his email. Just go to that episode. And he was really good at explaining things, so if you're still looking, check that out. All right, let's do some picks. Steph, what do you have?
Okay. So I have something. It's an article titled Scientists Rise Up Against Statistical Significance. It's very kind of powerful, rising up, but it's basically it's talking about how there's a lot of dismissing possible crucial effects, even though that they're not statistically significant or are near statistically significant, if we're disregarding them when there's actually a biological significance, and trying to parse that out between the lack of reproducibility in some of our science. And so using a p-value of 0.05 is very standard in our field. And so if it's larger than 0.05, it's not significant. If it's lower, then it is. And I remember, Vincent, you talking about this in virology. I mean, when we look at some of the effects we see with our cells and culture, significance really, if you see a difference, that biologically maybe matters more. So I'm kind of in between. I don't know if I'm rising up yet, but I think it's an interesting conversation to have. I don't know what your thoughts are on it.
Periodically this happens, right? I mean, I remember a couple of years ago, it was all about the tyranny of the p-value.
Mm-hmm.
Right.
People, and it's worth repeating periodically because some people are just tied to it. Oh, the p-value isn't low enough, forget it. But just look at the data yourself. Sometimes your instinct is really good.
Right.
Right. So I think that I saw this article too. It's really making the headlines, at least as far as science headlines go. Right. Maybe the rest of the world is not so interested. Cindy, what do you have?
So I have a blog post from a postdoc, Ephraim Rivera Serrano, who recently did a Twitter poll, and some of you may have taken this. He's quite active on Twitter at Naked Capsid, right?
Yes.
And so he asked PhD students and postdocs, have you ever considered changing careers midway through your training and or currently to do something completely unrelated? And so this is something that comes up. Do you quit grad school? Do you not quit grad school? Do you have what it takes? Do you not have what it takes? And believe it or not, 37% of the respondents said all the time they consider quitting or changing. And only 9% ever responded never. They never questioned it. And I fall into the camp for sure of the ones who questioned it. I don't know about all the time, but I had a crisis time period during my training, and I had had to decide whether I was staying or going. And lucky for me, I stayed and stayed committed, and I'm very happy. But yeah, I think it's par for the course that graduate students at some point will reach that and have that moment of crisis. But I think how Ephraim blogged about his personal experience and the struggles he faced and why he faced it, because he is a gay man and he is Latino, and so those were additional factors that impacted him and his experience and his upbringing in, I think it was Puerto Rico, and how that influenced how he felt when he was in graduate school and talking about imposter syndrome. And so I think it's a really powerful post. I think he wrote it very, very well. And I think everybody should read it, and especially people who are running labs, to understand that, there's things going on behind the scenes for people. Their life experiences color how they view things. And just the fact that there are at least a third of the graduate students and postdocs out there that think every single day, should I really still be here? And that's kind of scary, because this is a fun thing to do. It's hard. It's really hard, but it is a fun thing to do science and to explore and investigate and question and discover.
I wonder how, if you polled other trainees in other professional areas, what you get. You might get similarly high numbers, because anything that requires a lot of work and focus and is not easy and is associated with frustration, and as you say, behind the scenes things can affect your performance. I bet they all have high. I bet even medical students, a third of them would say, I don't
probably
do this anymore.
Yeah.
Well, I think what we can look at in terms of today is that people getting PhDs are seeing a lot of people never get jobs in maybe the field they want, or they're not seeing the support in terms of, okay, well, I'm not going to get an academic position. I got to go into industry, but I don't even know what that means. I don't know what that's about at all. And I think what this does signal is that the landscape of today is very different. And in terms of how, relating to PhD students working today, this blog post, I agree, Cindy, it's definitely worth checking out. And I'm just under I hear a lot of bad stories. I've seen a lot of just bad stuff go down in terms of like PI, PhD relations. And I don't think that you need to behave in a way that's nasty. We can all progress and do well if you just not yelling at people, kinder. I mean, I know that their science is challenging, but I don't know, just be kind. It's fine.
I think that part of that would be not having anonymous peer reviewing. You can hide, make nasty comments behind, a wall. I think for grants, for papers, it should be identified, and I think people would be less nasty if they were. I think this historical perspective would be interesting. If this poll had been done 10, 20, 30 years ago, it would be really informative, because Steph, your point is well taken that things change, but they may not. And people may have been unhappy for other reasons too. Maybe there were plenty of jobs, but for some other reason, getting a PhD would frustrate people. So, we can't answer it, but I think that'd be interesting. Anyway, what do I have? I have an article in Science called The Law and Vaccine Resistance. It's an editorial, actually, by Dorit Rubenstein-Reese, who is a law professor at UC San Francisco. And, we've been talking on Immune and also TWIV and other podcasts about the vaccine problems we've encountered. There's a big measles outbreak globally. I just read today 14 kids are dead in Ukraine from measles. None of them should be dead.
No.
There's an outbreak here in Rockland County, New York. And this is a real problem because they're all people who choose not to vaccinate their kids. And this editorial takes the perspective of, from a law perspective, what could we do? And I've always been interested in this because I've mentioned on TWIV not too long ago, we should start thinking about suing people who don't immunize their kids and then they spread infectious diseases to others and harm them. And so this is a short article, but it is very level-headed. And the main points are, what can we do about the current anti-vaccine movement? Well, she says states have extensive leeway to protect public health, and the courts have consistently upheld strong school immunization mandates. And she argues the states can tighten non-medical exemptions, where you just have to check a box now and get a non-medical exemption. She says valid medical exemptions are important, but it's less clear whether non-medical exemptions are appropriate. Some scholars are concerned that eliminating non-medical exemptions may generate resentment among parents and interfere with parental autonomy. Others, including professional medical associations, disagree because mandates protect children, and a parent's freedom to send an unvaccinated child to school places classmates at a risk of dangerous diseases. So, really, well thought out arguments. And then she says there are other legal tools that haven't been used. States could implement workplace mandates for those working with vulnerable populations. They could impose tort liability, civil law damages for harm when unexcused refusal to vaccinate leads to individuals becoming infected unnecessarily or worse, to a large outbreak. States could permit teenagers to consent to vaccinations without parental approval, and states could mandate vaccinations to enroll in institutions of higher education. I think these are all great ideas.
Yeah. Definitely.
And, we hear of people who say, I want a right to vaccinate or not vaccinate my kid. Well, I'm sorry, you don't have a right to infect others. And that's the difference. And that's what they're not thinking about.
Right.
Unfortunately. So I think it's really great. I don't know if it's behind a paywall, but it's really worth a read. I'm glad to hear from a lawyer on this because I think we're getting into that area now where we have to think about the law and how it regulates what's going on. Here in Rockland County, it's a big outbreak. A lot of parents were suing to let their unvaccinated kids go to school. They're not permitted. And the judge said, no, you can't. You have to be vaccinated to come to school. No doubt about it.
Right.
I think it was great.
Yeah.
And you've been doing a lot of, sorry.
Nope,
you've been doing a lot of really great public communication. I've seen they're on Twitter. You've been talking to different groups about vaccines. So that's great.
Yeah. Yeah. I was going to say, related to the suing part of it. Did you see the article about the child who got tetanus? $800,000 in medical bills. And the pain and suffering that child went through over almost 2 months.
It's terrible. Yeah.
And it's completely preventable.
And people don't think about this. The politician in Kentucky who gave his kids 6 or 9 kids, he gave them chicken pox.
I would love to have a conversation with him. What is going wrong with...
He said, yeah, they got a little sick, but they're okay now. Well, what if one of them had died?
Well, here, that's the thing. And also, we are still finding out what, especially in children, the antigens that you're exposed to affect the biology as you grow up. In terms of long-term consequences of viral infections, we're still finding out what could be going on. So that's just amazing.
Yes, we don't really know everything, especially when viruses get into the CNS. They can do things and have long-term consequences, right? We don't understand it all. So it's crazy for people who have a gut feeling that they want their rights to be making these decisions. It's just crazy.
And one of the other things that I thought about that senator in Kentucky, That's easier for him to say because he can have other people take care of his kids when they're homesick.
Yeah.
But
yes.
what about the
such a great
economic impact for most average households where when there's a child homesick, someone has to take off work and they don't necessarily get paid, and the business for which they work and the work is not getting done is also suffering? I mean, this is a huge economic impact to have children home ill with diseases for weeks that are completely preventable.
Mm-hmm.
All right. That is Immune number 18. You can find it at microbe.tv/immune, or you can subscribe on your phone or your tablet where you use a podcast player to listen. Please subscribe. Gives us the numbers of how many people are listening, and that helps us. If you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a few ways where you could, for example, give $1 a month through Patreon or PayPal. We really appreciate your support. Immune is Cindy Leifer from Cornell University. She's at Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langle is at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Thank you. This is great.
I'm Vincent Racaniello. You can find me at virology.ws. I'm PROFERR on Twitter. Music on Immune is by Steve Neal at stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.